Good morning, everybody. My name is Cojoc. I am from the National Research Council, Institute of Materials, which is located here in Trieste, but in Basovica, in the Area Science Park, where the electrosynchrotron and the Fermi laser, FEL laser, are located too. Our institute runs also some beam lines, and the labs are placed here, and I am leading the optical manipulation lab, which is placed here, more or less. Okay. So after the beautiful lesson you have heard, we will continue with optical manipulation, but in a different way, so with the use of light, but basically the use of radiation pressure of light to trap in 3D and manipulate particles. And then I will explain how we use this technique to run single living cell experiments. So the outline is in 3 points. I will introduce a principle. First, properties, then the particularities of optical manipulation for biological particles and living cells, and then we will go through some examples and we will try to learn something. Having as a motto this, I think, beautiful saying of Sidney Brenner: progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. Okay, in this talk by optical manipulation, we consider 2 things. We consider the use of laser beam in optical microscopy to trap, move, or ablate objects with size ranging from, roughly speaking, 100 nanometer to 100 micrometer. I say roughly speaking because they can be also smaller and bigger. Which tools are used for optical manipulation? Optical tweezers or laser scalpel. As we will see, optical tweezers allows trapping of micro objects immersed in liquid and manipulate them with up to 6 freedom degrees, while laser scalpel is a tool allowing ablation of micrometric objects with thin micrometric cuts. So it's the same as in normal surgery with normal scalpels and tweezers, but here we use light, so we do not have mechanical contact. This is the main property, or the main difference. I have to specify that the presentation is not different from the slides that the materials that I send. They are just less. So, because otherwise I do not make it. So what is an optical tweezers? It's a laser beam focused through a high numerical aperture objective, allowing to trap particles in 3 dimensions. So if we consider the microscope objective and the illumination path now, and the sample plane, and then here we go in green, we go to the imaging system, we insert a dichroic beam splitter, which allows us to direct a laser beam to be focused onto the sample plane. And if there we have microparticles, due to the interaction of the light with these microparticles, one microparticle can be trapped in 3D, so kept like with the tweezers. So schematic, we have this situation. We have the Petri dish, whatever, the sample, and microparticles. Here, for simplicity, there are beads, and 1 bead, and the laser beam focused inside the sample chamber or sample cell, and the bead trapped. If we want to move the particle, we have to move the tweezer. That is, we have to move the focus of the beam, and the particle will follow this. Here you have some numbers. So the force exerted on the particle depends on a dimensionless efficient coefficient, which is a function of the geometry of the material of the particle, of the power of the laser, and also of the refractive index of the medium, and of course, the constant light speed. So as an example here, we can see the trapping of a red blood cell. So we move the stage. Here is a trap, and you see a red blood cell is trapped and rotated like this. So it follows the axial, the optical axis. So the disc shape makes that the cell is trapped like this. A simple picture of the force exerted by a ray of light, I took it from Arthur Ashkin, who is a pioneer of optical tweezers. He begins this work in early 70s in Bell Labs. If we consider a ray of light, so I assume that here there are more biologists than physicists or engineers, so we make it not simple, but we try to make it in such a way that it makes sense. No? So a ray of light, we know that light contains photons, and photons are characterized by energy. But we have also linear momentum associated to the photons. No? And as a consequence, so we can say that if we have a ray of light, we have a number of photons there, and this ray of light interacts with a bead, with a sphere which has a refractive index higher than the refractive index of the medium in which it is inserted. The ray of light is bent, and again bent, and then if we apply the conservation law of momentum, we have the variation of the momentum which gives rise to the force. No? So the force of the photons exerted on the sphere, and then we have also the force exerted by the sphere on the photons. For many years, I had difficulties to explain to biology environment how an optical trap works, until I had a very good student, biotechnologist, Elisa Deste, who gave me this explanation, and from that moment I continue to show this. So here we have the laser beam now, and the intensity profile of the laser beam, which is Gaussian, is not uniform, okay? And we have a high numerical aperture objective. Everyone knows what is a high numerical aperture objective. If not, we know that it is the angle, the high angle, this angle here. And in principle, if you have the same focal length, you need a large objective, a large diameter of the objective to have a high numerical aperture objective. I will try to explain why we need a high numerical aperture to trap in 3D. Okay? So if we consider rays coming near the optical axis and interacted with the bead, we see the force having this direction. So it is with the axial component pushing the bead away from the focus, and an orthogonal force direction which tries to move the bead away. But since we have symmetry, we have these two forces trying to move from the optical axis compensate, and we have only the force pushing the bead away. In fact, this is what happens with a normal low numerical aperture objective. The beads are attracted towards the optical axis and pushed away. If you consider the rays coming from high angles, large angles, the force has a component, the force has this orientation, where we see that there is a component trying to bring the bead back to the optical focus. The same here, the same here. And then we have the equilibrium when the bead is in the trap. So I think Elisa was right and pretty efficient. So Arthur Ashkin is here in the Bell Labs with his technician, Zejic, with who they did very, very nice work in the 80s. And he published only one book, but very good book in my opinion for who is interested to go deeper in this field. And even the cover is beautiful. What you see here is levitation, particle levitation. So it's a laser through a low numerical aperture objective. You see that you do not have a high convergence of the laser. It's focused into a capillary. And this is a bead of about 50, 70 micrometers. So with an eye, we do not see it, or with a photographic camera. What we see the scatter light. The laser comes here, and the light scattered by the particle, we see it. And the particle, the laser compensates the weight of the particle, so it's levitated. What is more difficult to be seen is this piezoelectric ceramic, which at the beginning shakes the sample to detach the particle from this surface, because the forces are van der Waals and they are small, nanonewton, but the forces to maintain in equilibrium in levitation the particle here are piconewton. So the laser is not able to detach the particles from the surface. You need something else, but once the particles are detached, they are, so it's, I think, beautiful cover as well. So some properties. What type of particles can be trapped? As material, we have dielectric, polystyrene, silica, but also metallic, gold, silver, copper, biological, cells, micromolecules, intracellular structures, DNA filaments, so huge variety. Low index. By low index, we mean when the particle has a refractive index lower than the refractive index of the medium. For instance, the bubbles. The bubbles now they have gas inside, and they have a lipidic shell, and they are inserted in water. For instance, the ultrasound contrast agent bubbles used in echography are like this. As shape, also spherical, cylindrical, arbitrary. Now, do not think that all of these types can be manipulated as easy, so it's the same. So some are more difficult, we need some tricks, but in principle, you can manipulate all of these. As size, important thing, we have also a very wide range, from 20 nanometer to 20 micrometer. As forces that can be applied and measured, the range is between 0.1 to 100 piconewton, which is, by the way, the interaction forces between cells, between macromolecules and cells and so on. So we can play also with mechanics to induce, to mimic behavior of the cells. Some examples, biological sample of trapping. So there are some questions. One question is related to the intensity of the laser, because the intensity of the laser at the focal plane is huge. So does this damage the cell? If yes, which is the level of the damage? This is also something, I am an engineer, so this is also something that I learned in time, making presentations, because at the beginning when I asked engineering environment or physics environment, did you damage the cell? No, because the cell is still alive. Okay, if I ask you, I hit you, and I do not kill you, maybe I damage you, but the level of, is right, Alberto? Because the exception of maybe I forced a bit, but the exception of terms coming from biology is normal to be different in engineering and physics, but at the moment they should be equalized, no? They should be the same. So care should be taken what is damage. Maybe it is damage from one point of view, but from what I am interested to study, it is not, and it works. Okay, then the fact that biological samples have very, very arbitrary shapes, and if this prevents trapping. So, of course, the answer is no. Okay, so the first proof of trapping of a biological sample was given by Ashkin and Zizic in a paper in Science in '87, very nice. It is very nice in my opinion also because they use laser scattering, so they observe the sample from the lateral side, and they, by scattering, because at these sizes you do not see the sample, you cannot image, it is below the resolution limit of the microscope, but you can use the light scattering to know that you have the sample there. And moreover, they were able to detect the particle and their orientation with respect to the optical field orientation, and also to know how many particles, and from the scattering to know also how much it is damaged. And they increase the power of the laser, and they observe when they kill. So, simple but useful. A simple diagram, a simple graph here to show why the region of wavelengths that it is used in optical trapping is infrared. Because we have a good compromise between the absorption of light by the components of the cells, biological components, and the water. So if we go toward higher wavelengths, we have absorption of water, and we have a lot of water in cell. If we go toward low wavelengths, we have a lot of absorption by proteins, by components of the cells, and it is not good. This is why the compromise is use infrared lasers. So here they are not very beautiful for our days photos, but they are relevant because it's the first trapping of cells, yeast cells, with infrared, which is again a Nature paper. This, in our days, is no more a paper, not Nature to speak. But it was very important at that time. And then also another important paper demonstrating the trapping inside the cell. So of subcellular components here, there are cytoplasmic filaments. No? So we have the nucleus, we have the initial position and the final position. So we have a tether inside the cell. So what if the absorption coefficient and intensity of the laser are high? We have laser ablation. So instead, we damage, but we damage because we want to damage, and we damage locally. We cut there. So the inventor of that is Michael Burns. There is a good, very good review in Methods in Cell Biology. So the idea is that the laser ablation occurs when the energy delivered to the sample is higher than the breakdown energy threshold of the irradiated material. So this depends on the laser wavelengths, on the pulse duration, and on the lens numerical aperture. So the first paper was again a Science paper, and here in these pictures you can see a single stress fiber of a rat endothelium cut by a 1 pulse of a YAG laser microbeam here, and then regeneration of the stress fiber. I do not have time in the material that you have. You have more information about groups from IIT, different groups using laser ablation on neurons, for instance, to cut neurites and to study the regeneration of axons and neurites and so on. Okay, some examples. We go back to red blood cells that we showed at the beginning. We know the shape, we know that they have an important function, we know that they do not have a nucleus, we know that the cytoplasm mainly consists of hemoglobin and they have a limited metabolism. And there are a lot of interesting studies, for instance, for in malaria or for other diseases involving red blood cells degeneration. So I do not go too much into details, so this we saw already. What I want to show, I want to show a simple configuration with two objective microscopes here, one vertical and one horizontal, and a capillary, a very thin capillary, 100 micrometer. And what you see here are the two views from lateral and from the bottom, or from the top as you want, of a cell manipulated with two traps. Okay? So this is a capillary wall, and you see the red blood cell. Okay? It is rotating. It is rotating. Here is the lateral view, and this is a top view, which is at a higher magnification. So it's clear? It's clear. So this can be a useful tool for manipulating the cell and for stressing also the cell. For instance, another group from Spain, they did a very nice experiment stretching the cell and measuring the Raman the Raman scattering. So the signature here is part of the signature, the change of the signature as an effect of the mechanical stress. So they established the relationship between chemical binding and mechanical force in oxygenation cycle of the hemoglobin structure. Another application is to surround a cell, and we tried this also with Federica, with some people from ICGB, a cell with bacteria in known positions, and then to maintain that configuration by fixing the gel, but not killing the cell, and observing the cell after incubation, some day, 2 days in incubation, observing the cell interaction with the bacteria. This is another type of manipulation. Here already you see using multiple optical traps. And you might wonder how you obtain multiple optical traps. You can obtain shaping the beam, the laser beam, trapping beam, as Matthew said, with a video projector-like device. It's a spatial light modulator. So with a device, instead of getting one trap, you obtain multiple traps, splitting the input beam in different beams that you can configure spatially in a dynamic way. Okay, so another application, a recent application from our group is on extracellular vesicles, which are circular membrane structures released by most cells, which represent highly conserved mediators of intercellular communication. So it's an object of very high interest. And here the goal was to see if we have adhesion of extracellular vesicle obtained from glial cells on astrocytes and microglia. So in the movie what you see that we place the vesicle on the cell, then we switch off the laser and we look what happened with the vesicle. Maybe here is clearer. So we have the vesicle. We position it on the microglia. Then we switch off the laser. Then we follow, and we see that not only the vesicle adheres to the cell, but also moves on the cell. And then we look how it moves. It's a blue track. At the beginning, it moves around the same position. As you see, it moves quite a lot, but not the distance to the original position remain reduced, and then it goes towards the cell body, and this is repeated for more elements. Then we repeat these experiments also with neurons. So we place these extracellular vesicles on neurites, and then we let them interact without the laser light. And we see that the vesicle moves always on the neurite toward the cell body. A lot of other investigations are necessary to understand why, which is a mechanism of this movement, which is a mechanism of interaction, which is a mechanism of endocytosis, because they disappear and they are absorbed by the cell. But is so a small part of the work. With optical tweezers, we can apply and measure small forces. So the idea is that in an optical trap, the optical trap can be characterized by a trap stiffness. We can assimilate it with a cantilever of an AFM. So I do not have time to enter into detail how we measure the spring constant of the trap stiffness, but assuming that we do this in a step which is called calibration, then if we apply an external force on the bead in the trap, from tracking the displacement of the bead in the trap, we can measure the force. Because we have the spring constant, we measure this, so we get the force. So in our lab, and not only in our lab, in many other labs, there is an interest to compare the results obtained by probing cells, living cells with AFM and with optical tweezers. Why? Because the forces and the range of forces and stiffness are very different. And there is the result, the elasticity, the results obtained from probing are also different. And the question is, which method is better? Or maybe both are good and maybe they are complementary. So I do not enter in too much detail with the setup. It is quite simple. So it is, this is a microscope. This is a sample. We need a nano piezo to position the sample and to calibrate. We have the infrared laser beam, and we have the optical path of imaging. No? And in addition, we need something to detect the displacement of the bead with nanometer precision. And this something is a quadrant photo detector on which the interference pattern formed by scattered light by the particle on the detector, so on which the interference pattern is formed, and we can measure its displacement and associate with the displacement of the bead. So the principle is the same as in AFM. So we have the bead in the trap, we move the cell first down, the stage down and the cell, and then up, we intercept the bead and we indent the cell. From these regions, B and C, we have the indentation and the retraction regions. So from these regions, we can detect we can measure the force and the displaced indentation. And from this, applying the Hertz model, we measure the elastic modulus. So we measure the elasticity of the cell. Okay. What we do is this. We try to compare the elasticity of different types of cells. In this case, we analyzed breast cancer cell lines, non-neoplastic, low metastatic potential, and high metastatic potential. And we compared the results obtained with optical tweezers and with AFM. And we observe, and also other groups observe that the metastatic cells, they are softer. Okay? They are softer than the other two cells. And this is the same for AFM and optical tweezers. This means that both techniques work well, even if, notice how big a difference is between the elastic modulus measured with optical tweezers, tens of Pascal, and with AFM, tens of kilopascals with this technique. And this is explained because we have different loading range. The cell answers differently. The point is that you stress it in the same way to compare different types of cells. So there are no absolute values about the elasticity of the cell. Relative values, yes. Relative values between different types of cells, but also relative values as a function of the environment condition. Because the cells interact with other cells, they interact with the substrate, and actually what we studied how much the stiffness changes when the elasticity changes when the cells are in contact or not. And the surprise was quite big. The metastatic cells get stiffer when they are in contact. Then again, one says, okay, can be this a criterion for diagnosis? In my opinion, not. Or it should be taken with tweezers. Another group analyzed the interaction with, analyzed the interaction with the substrate. There is in the material, but I do not have time to enter into detail. Here is a nice experiment where they analyze the adhesion. So the adhesion between a bead functionalized with scadarins and the cell when the surface area, the area of the pattern on which the cell is plated changes. And there are also surprises there. Surprises in the sense that nothing can be given so easy as known or found. So the main message here would be, in my opinion, yes, there are different methods to investigate cell mechanics, but we always should take into account the environment, the condition of measurements, before we express conclusions which refer to diagnosis or so on. Cell-to-cell interaction. So here I will speak about biomechanochemical signals, and I will focus on neuronal field and interaction between young neurons, between growth cones of neurons during the development. And I start from this movie in which you can see how two neurons speak each other in 2 hours from a morphological point of view. As we can see, probably there are also other, the language includes also other stuff, no? Because there are chemical signals transmitted each other. You see that there are a lot of contacts, there is a displacement, they seem to like each other, and to connect, because they are growth cones, they are at the beginning of the network formation, which is very, very important, the first 2 days, when it is very important how the neurons connect each other. So this is why we study, we work on this together with a group in CISA and in University of Trieste. So the goal as a method is to create physiological inspired experimental conditions for instance, to mimic one of the two neurons in the previous example by using functionalized microvectors carrying the stimuli and manipulate them to stimulate the neuron at specific sites. So you know that in classical bus administrations that you use in most of the cases in the lab, to change the chemical condition of the cell, you pipette molecules that they diffuse around, they get to the cells, and then you look at the reaction to some cells that you consider that they are good. They show what is expected to show. In any case, this situation does not reflect the physiological conditions where gradients are very important. You saw also in the previous presentation that moving the laser, so creating basically gradients, induce a reaction of the cell. Because if you just shine, I guess, all the cell with light, the situation changes, but is change and then remains fixed. There are the gradients which are spatial gradients and temporal gradients which are very important. There are different ways to introduce gradients. One approach is micropipette-based assay, but this works with quite big amounts of molecules. Then there is the optical uncaging of cage molecule, which is very nice because it's very high resolution approach, spatial resolution approach. Unfortunately, there are not too many molecules that you can work with this. And also this approach that we use to encapsulate molecules inside liposomes and to bring them with optical tweezers near the cell and then break the membrane of the liposome by photolysis and release the molecules locally to the cells. It was introduced previously not in neuronal cells, we introduced in neuronal cells. So now I will show you the use of two types of vectors. One microbeads and one is lipid vesicles. I will speak about two types of vectors and I skip basically one very nice vector that I just say two words, biopolymers beads, in which you can encapsulate drugs and you can use them to release and to create molecules and to release, to create spatial gradients, spatial and temporal gradients, and to stimulate cells. I will speak here about microbeads and liposomes. Functionalized beads, that is silica or polystyrene beads, which are coated with carboxylic group, and you can easily functionalize them with every type of protein. And this is one type of vector. Another type of vector I just said are molecules encapsulated in liposomes. The difference between these two is obvious. In this case, you cannot detach the molecules from the surface. In this case, you release the molecules, but here you have a better control, spatial resolution, you know where you apply. Here, this is relative. So, how it works? So, this is contact with beads, and this is with liposomes. You need an UV pulse to break the membrane of the laser. And I will give you an example with microbeads. Is in collaboration with Enrico Tongiorgi and Gabriele Bai from Brain Center, University of Trieste. And in these cases, we use functionalized beads with carboxylic group, and we coated them with brain-derived neurotrophic factor. And the goal was to see if a single bead is able to stimulate the neuron. We knew more or less where we should apply from previous experiments with beads in bulk. where we should apply the signal. And we checked that this works with 4 experiments. I will show you one. This one with an increase of calcium in the cell body and stimulated neurite. But we demonstrated also that the receptor track B was activated, and that the C-phos translocation in nucleus was induced, and additionally that we have an increased motility of the neuron due to this stimulation. So the calcium increase, you can see here the neuron, the cell body, the neurites. First we apply a bead with a control, with BSA, coated with BSA, and we look at the fluorescence, and we do not have a change in fluorescence. And then we apply the BDNF bead, and we look the increase of fluorescence in cell body. And we notice also an increase of fluorescence in the neurite. So this repeated many times and compared also with a case when you have BSA bead to cancel the possibility that is a mechanical stimulation. And now we move to liposome. So this was an example. You have possibility to see other experiments and other examples from the materials that I gave to you and from literature. Liposomes. So the liposomes are spherical vesicles ranging from 50 nanometer to 50 micrometer, and they are called giant, large, so on. So they are lipid vesicles, basically. Liposomes is a term more for 100 nanometer and small vesicles. They have a phospholipid bilayer membrane, and they have an aqueous core. So we speak about this vector, but we might consider also this situation, which is interesting. We do not apply it in our lab, but it is interesting. What it is also interesting to notice is that a liposome of 1 micrometer diameter, we like 1 micrometer because we can manipulate, we can see in the microscope well, filled with 1 nanomolar solution, contains 1 molecule in mean value. No? So you stimulate a cell with 1 molecule. Then the important thing is this is enough or not. This can be seen. So to have the picture of how the release and stimulation gradient arises, look at this figure. So you have the liposome, here you have the cell, you have a point on the cell at a distance r, and the question is, once we know the size and concentration of this vesicle, and we release the molecules from here, which is the concentration here, okay? And which is the distribution along the cell. And for this, we adopt a simple model diffusion from a source point here. And as you can see here in this graph, we have a spatial gradient and a time gradient also. No? So basically, as time gradient, we see that after a few seconds, the situation stabilizes. After 5 minutes, for instance here, we have a spatial gradient from 10 to 50 micron created, which remains on the cells. This distance is measured from the liposome. Okay? Playing with these gradients, with the concentration of the molecules inside the liposome, we can scale down the number of molecules that will get to the membrane receptor. Okay? One of the examples is related to the focal stimulation of hippocampal neurons by PRPC, so the cellular prion protein, which you know better than me that it is very important because it is present in almost all the cells, and its functions are not yet known, all of them, and also because of its bad counterpart, let us say, the prion. No? So we encapsulated PRPC, recombinant PRPC in liposomes and delivered to neurons, and we found that PRPC behaves as a guidance molecule, which was not clear very well before. That also membrane PRPC is required for the extracellular PRPC to bind, which means that PRPC might be the receptor of itself. Then that the full length of PRPC is required to have the guidance function, and then that the growth cone growth guidance is modulated by the concentrations that we have in that. This is a collaboration with CISA, with the group Prion Biology, the group of Professor Giuseppe Legname. And here I show you an example, a sequence of images with PRPC and with a control, liposome with a control with BSA. And the movie, I hope that you can see it. The liposome is here. We released, you see that it was broken. We released the molecules. And in 8 minutes, you see how the neuron grows toward the initial source. Do not be scared about these guys traveling here like crazy. They are in different planes, and they are, in any case, they do not influence the growth in this direction. So we noticed a significant growth induced by PRPC delivery. And as I said, we noticed that PRPC knockout neurons do not respond to the stimulation with PRPC. The paper is under revision. No, I do not say where. Okay, so as I said, we used, so this is a good method because it can be applied with any type of molecules, basically you can use. In collaboration with Vincent Torre from CISA, we played with other guidance cues, with netrin, with semaphorin, and for instance here you see the liposome and the neurite getting to the liposome for because netrin sometimes is attractive, no? Sometimes it depends on what it finds to photolysis, and you see these are okay. Then by now we saw morphology and so morphological effect, and we want to see a bit more in detail what happens. So for instance, what happens with the signaling pathways, and in this project with CISA, we want to see the activation of the GTPases of RHO-A and CDC42 when we deliver semaphorin locally. Because it is not known if we have here a role or not in this pathway. Because of this, and it is also not known the spatial very well how the spatial temporal activation of the GTPases work under stimulation as a result of stimulation locally and with different concentrations. Okay. So for this we used FRET as and FRET probes, of course. And for who doesn't know about FRET, is first resonance energy transfer, and it is a fluorescence method, but where you have a donor and an acceptor, and you excite the donor, And due to non-radiative energy transfer between donor and acceptor, when these two molecules are very close, only when they are very close, closer than 10 nanometer, you get an increase of the fluorescence in acceptor. Okay? So it is a very good indicator, for instance, if you have a protein and you want to study the conformational changes of this protein and you have a probe like this, it is very good because it indicates you this. So we use two types of probes, one intermolecular and one intramolecular. Actually, the results that I show you are with intermolecular only because we have big difficulties with intramolecular in terms of toxicity and efficiency. So efficiency in transfection means that you have a lot of good cells, transfected cells, a good percentage, but they should be also alive. I mean, alive like they should be. And we had problems. Now it seems that it works. With this is easier, but the point is that you do not have a stoichiometric control. So there might be more molecules interacting with one, donors with one, and you do not know this. So here we use sensitized FRET that I will explain in a moment with the optical setup. So the optical setup includes a microscope with the fluorescence. The only thing is that the exit port of the microscope, we add an optosplit device which allows you to separate the two channels. It's like in calcium ratiometric, for instance. So you have the fluorescence from the donor and from the acceptors that you can separate. And you project, in our case, we project on the two halves of a camera, but you can use also two different sensors. And then these two signals allow you to analyze the FRET signal, so to detect if you have FRET and when you have it. So this is the fluorescence part, then the excitation. Okay, here we use the mercury lamp, and then here this is a laser, the pulse laser for breaking the liposome, and the infrared laser for manipulating the stuff. Okay, so the results that Federico Isepon mainly and Luisa Napoletano, they were the main players in this paper, obtained raw dynamics. So we open the delivery of semaphorin from liposome. You have here the cell, and here is a liposome, and this is a sequence, and here is a movie, basically. So you have the border of the cell at the beginning, and then what you see is the signal, is the ratio signaling indicating the raw A activation. Okay? So what we are doing now, we superpose also the morphology, so the DIC imaging, which at the beginning was not very successful. So here in this graph, you can observe the edge retraction and the level of the activation, or rho A. Then for CDC42, actually here it is something which here you see the stimulation, and you see a part of the cell which retracts, because CDC42 shows me this, but also a part which you have some protrusion. Okay? And this might be related somehow also with the mechanisms that Matthew showed before. What was relevant here, so observing the two signals in two regions, we have the edge growth, so we were interested where the protrusion was obvious and where we had retraction. And we observed some waves behavior of the CDC42. Useless to say that I am not very prepared with the biological part, but I think you got a flavor of about what can be done, and so in conclusions, we can say that optical tweezers allows gentle trapping and manipulation of living cells without mechanical contact, allows probing forces in the piconewton range, complementary to AFM, where we have tens of piconewtons, hundreds of piconewtons, and nanonewtons. Optical tweezers allows us to handle vectors carrying active molecules to stimulate cells. And with this, we showed local stimulation by coated beads, which is simple and extremely flexible. Any type of protein can be cross-linked on the surface. And field liposomes are flexible as well, and the released molecules can interact freely with the cell. So, and what it is from the technical point of view important is also that optical tweezers can be easily integrated with other microscopy techniques. So we have a platform where we can insert more. So in conclusion, it is good that we manipulate, we see what we manipulate and manipulate what we see. No? Here I have to thank many collaborators in time, and to you for your attention.